Hello class, this is Mr. Allman. I wanted to take you through a little bit of review of cell respiration. This will probably take a couple of posts, or sorry, I should say a couple of videos, because I wanted to make sure we circled back and hit a little bit of review in terms of energy and enzymes, because those are really significant concepts in terms of understanding respiration, which is ultimately a biochemical process. So first, a little bit of theory and practice related to how these reactions work. Looking at a couple of laws that are significant here. The first is that matter and energy can't be created or destroyed. So we can't make atoms, we can't destroy atoms, we cannot make energy, we cannot destroy energy. That's not going to happen in any living thing. However, matter can be brought in, energy can be brought in, and they can be transformed from one thing into another. So if we take a look here, you can see the bear. We have the little red arrows representing heat. So that's energy leaving the bear, lost as heat. And we see a couple of the other things that are leaving, the carbon dioxide and the water. Those are both waste products of respiration. Both of those are leaving the bear. Now, ultimately, energy comes from the sun, and that energy is going to be trapped through the process of photosynthesis in plants. And the chemicals that are made, like the glucose that's made in photosynthesis, will store energy that can be released later on. Now, chemical reactions can be divided into two types. There are exergonic reactions, and those are reactions that are going to release energy. Sometimes they happen spontaneously, but in many cases they still need some type of trigger. And any molecule dividing, which is known as a catabolic reaction, is one that is going to be exergonic. Now, as you can see in the graph here, energy is on our y-axis, and the reaction, the time, is ultimately on the x-axis. So you can see the energy present in the reactants is very high. Now, there needs to be something to trigger a reaction, and the products that are formed are usually smaller and simpler. So we usually have a larger molecule being broken into smaller molecules, like, for example, a glucose molecule being broken into carbon dioxide and water atoms, molecules. And so there's going to be that difference there is the amount of energy that is going to be released. And that energy can be captured and stored in other molecules to be used by the cell for other purposes, to do more work. Other reactions are going to be endergonic. Endergonic reactions is where energy is required. It's not spontaneous. It needs something. It needs energy to be able to trigger it to happen, and it usually needs something like an enzyme to catalyze it. These we define in biology terms as anabolic, molecule building reactions, because an anabolic reaction is one, if we think about what, for example, an anabolic steroid does, is help to build particular molecules in the body. So someone who takes anabolic steroids is going to have a greater ability to build additional muscle mass because of the work that those steroids are doing to allow those molecules to be built. So you see, you start with simpler reactants, and you are creating more complex products. Like, for example, when a plant takes CO2 and H2O and combines them together to make a more complicated product like glucose. So here energy is going to be required, and in the case of photosynthesis, sun is the ultimate source of that energy. Now, another law that governs how energy works in the universe is that in any closed system, it's going to tend towards a state of maximum entropy. And so this is describing how ultimately everything is going to even out. Like, for example, diffusion. Things are going to go from a higher concentration to a lower concentration. And the same will be true for energy. If we think of the example of heat. So if you have one room that's hot and another room that's cold, and you open a door between them, the heat is going to travel from the place where the energy is in the hot room, and it's going to even out with the cold room, and they'll ultimately end up as the same temperature. Now, life is going to be highly ordered. So some examples here in both plants and animals, you see that the very distinct geometric shapes. That's a sign of order, and that's happening because of what life is doing with energy. It's taking energy in, and it's using that energy to maintain and increase order, which is and so because of the fact that the universe tends towards a disordered, random state, a constant flow of energy needs to come in order to maintain that order. And so for animals, it's in the source of food. So you see the bear eating the fish. It needs the fish because the energy that the fish provides is going to fuel the work that the bear needs to do to make sure that that order and stability will be maintained. You can see energy present here in the jellyfish because of the proteins that they produce that will glow or phosphoresce. That requires energy. And every living thing needs that energy in order to maintain that order. Now, another little bit of review relates to ATP. So ATP, which stands for adenosine triphosphate, you do need to know the name. You need both the abbreviation and the full name, and you need to know the basic structure. We have the adenosine, which is present here. Adenosine is a type of nucleic acid, so it's the same A, chemically related to the A that is one of the nucleotide bases. Then we have the sugar here, and so here's a ribose sugar, and then we have 3 phosphate groups. Each phosphate group consists of a single phosphorus and a set of 4 oxygens that are bonded to that central phosphorus. And this is a short-term energy storage molecule for the cell, and as it says, tens of millions are going to be used per second per cell. So it's extremely common, extremely important, because it's the way that the cell is really moving energy physically from one place to the next. Now, the reason why it's able to use that is because the bonds between the phosphate groups are relatively unstable. The cell can pretty easily snip and release one of the phosphates away from the other 2. And the fact that is an easy bond to be able to break allows us to utilize that difference, that change in energy that I mentioned at the beginning of this video. So much more energy is going to be released when the bonds are broken than putting them together. So here you have the full ATP. You got the adenosine, you got the ribose, you have the 3 phosphates. If we clip off one of those phosphates and release it, there's going to be a little bit of free energy that can be harnessed by some other molecule or some other part of the cell that needs that energy. That process is known as hydrolysis because some water is going to be used up as a result. And ADP is the name, adenosine diphosphate is the resulting molecule. So either when energy is going to be required, any process in which energy is required, ATP is going to be broken down, one of the phosphates will be removed. A couple of examples where ATP is used is the transportation of solutes. So this is something, for example, like the sodium-potassium pump. If you have a carrier protein that is responsible for transporting molecules into or out of the cell, for pumping them, in this case, out of the cell, that ATP can be split, the phosphate can be added to that protein. When the phosphate that was released from ATP is attached to it, that will cause a change in the shape of the protein and releasing that solute out of the cell. A second example is going to be some mechanical work, like an example of moving things along the cytoskeleton. So if the cell needs to transport something from one part of the cell to the other, or if the cell needs to just release it entirely, you can see that ATP is needed when ATP will attach to a protein that's attached to the cytoskeleton. Every time the ATP is attached to it, it causes that protein to move, and it moves a little bit further along that cytoskeleton. And then you have to just repeat that process over and over with more ATP to move this vesicle in the direction you need it to go. So metabolism as a word refers to all of the chemical reactions that happen inside the cell. So ATP is central to this because every time food from any source is going to be broken down, that is catabolism. It's producing more ATP and producing some water. And then when that ATP is broken back down and that phosphate is released, that's going to release some energy, and that ADP that results from it can be reused back in that catabolic process. So you have this cycle where the ATP is constantly being reused, the phosphate is released and then added back on again. So the whole purpose of respiration is to be able to take this ADP and this free phosphate, to take those and put them back together. And the process of digesting food inside cells is ultimately what combines these things back together. So a little bit on enzymes, just to close this particular video. So enzymes are proteins. They act as catalysts. Catalysts are molecules that help in some way to be able to reduce the amount of energy that's needed for a reaction to happen. So you can see the black line here shows an enzyme, shows a reaction. The amount of energy that's needed you see is much higher if there's no enzyme. If we add an enzyme in, that red line is a little bit lower, and you see that then it's a little bit easier for the cell to be able to release that energy. So usually how they do it is that enzymes will physically combine. One note here, the ASE ending, that's going to be the signal in terms of the word of how you know that something is an enzyme. So enzymes usually interact with the thing that they are trying to change. So the substrate is what's going to change as a result of the chemical reaction. So here you see a couple of reactants. You see they have a particular shape. The enzyme is usually much larger, and it usually has some spot on that enzyme itself where those reactants will physically fit in. Once those two things combine there, they form what's called the enzyme substrate complex, and they will combine or change in some way and then be released by that enzyme. The part of the enzyme where those things will fit in is known as the active site. And the enzymes are reusable, so once they've done the job once, they can repeat and do that job over and over and over again. And what they're doing is making it usually a little bit easier, maybe stretching the bonds for one that needs to break, or forcing together a couple of molecules so that it makes it just a little bit easier for those things to chemically change in some way. Now, these things can be regulated, so enzymes can be turned on and enzymes can be turned off. You can take a look at the Prezi for a little bit more information on the types of interactions that happen, both competitive, non-competitive, and then finally allosteric. And these three types of interactions will serve to either increase the ability of enzymes to do their job or to decrease the ability for enzymes to do their job. And that's particularly important because we don't want enzymes to be continuously going through a particular reaction. A cell needs to be able to control the specific times when the enzyme will work and when it's not going to. Finally, in terms of organization, all of this is going to be compartmentalized specifically into the mitochondria. So the mitochondria represent those little pill shapes within the cell. There's always a lot of them within cells. The heart cells are known in particular for being packed full of mitochondria, and any cell that needs a lot of energy and needs to do a lot of work is going to have more mitochondria. Here you can see the blown up image of a micrograph showing the little squigglies inside there are the double layer. That's the inner membrane of the mitochondria. So you have an outer membrane along the outside, and then you have an inner membrane that is within. With enzymes, and the environment influences how well enzymes do their job, because enzymes are proteins, and proteins that are held together by some very weak chemical forces. And so changing the environment will cause a change potentially in the shape of the protein. So as you can see here with a couple of examples. So for a typical human enzyme, its best temperature is going to be 37 degrees Celsius, which matches up with body temperature. Now, if you get a little bit too far, if it gets a little bit too hot, like if you had a severe fever or if you had overheating, as the body temperature rises, those proteins are no longer able to do their jobs. As you get colder, similarly, proteins are less able to do their jobs. And so body temperature matches up with the temperature that our proteins need. If you're a different organism, like with thermophilic bacteria, a type of archaea bacteria, they have a higher temperature where their enzymes will be working optimally. Here you can see a couple of enzymes in terms of digestive enzymes and their pH level. So you can see a stomach enzyme, pepsin, it works best at pH of 2. And because the stomach contains a lot of hydrochloric acid, it makes sense that the enzyme will work best at that temperature. Whereas the enzyme trypsin, which is in the intestines, which do not have acid, they have a much higher, a little bit above neutral, pH 8 is the best for them. So ultimately, three things are important in terms of enzymes. First, temperature. So you can see a range here of temperatures, increasing gradually and then decreasing very rapidly above body temperature, which is 37 degrees. For enzymes in general, for pH, usually neutral or slightly basic is the most optimal for them, with the exception of those stomach enzymes as another example. And then finally, the concentration of the enzyme, the substrate, and cofactors. So the enzyme, you need a higher concentration of enzyme, and substrate, a higher concentration of substrate in order to get that reaction to happen faster. So those are the three things ultimately affecting how well the reactions may happen in a body. So altering the pH, altering the temperature, or altering in some way the amount of substrate or the amount of enzyme, those are going to serve to disrupt those reactions in the body, because as proteins, they will possibly denature, meaning that tertiary and quaternary structure will come apart and they're rendered non-functional and they're just not going to work anymore. There's feedback. We won't go into the details on this, but there are ways in which, for any chemical reaction, in addition to inhibiting the enzyme, there should be some other ways in which the cell can signal to say that, for example, we have enough of a particular product to stop making more. If a company is making, if you look at the Wrigley gum company, they are producing chewing gum, and if the shelves are all filled with gum, then that's feedback back to the factory saying you need to stop. We have enough gum, we don't need any more. And the cell works in a similar way. It has ways of sending information back to the beginning of these processes that are happening and letting the cell know when to start and when to stop those processes. So that is the background here on the theory and practice relating to some of the issues that we're going to cover in the process of cell respiration. So take a look at the second video for some details on the actual process itself.